I welcome you to this video tutorial. Today we will discuss acetyl CoA formation and regulation of PDH complex. In this video is in fact the continuation of the previous video. In the previous video we have seen how starch and sucrose is degraded, and we have seen it up to the level of pyruvate or pyruvic acid formation. That means we have completed the steps of which are up to the formation of the glucose converted to pyruvic acid through a multiple step process, which is 10 steps, which is called as the glycolysis. End product is 2-pyruvic acid, and now we have to see the second step, that is how this pyruvic acid will be converted to acetyl coenzyme A. Of course, with the utilization of an enzyme complex, and then this whole process is taking place within the cytoplasm, within the cytosol, and now we have to see that this process which is taking place in the cytosol, there is a formation of pyruvic acid. Pyruvic acid is to be transported to the matrix of the mitochondria, and then we'll see how the third step, that's the conversion of this acetyl coenzyme A into citric acid cycle, and then the next step will be how the reduced coenzymes produced from this will be fed to the electron transport chain, and then the electron transport chain, what will be the last step, that is the oxidative phosphorylation. So today our focus will be on this point. We have seen up to this point that the glucose is formed and it is converted through glycolysis, results in formation of an ATP, which is a substrate level phosphorylation, and reduced NADH. And of course there is a formation of pyruvate. Pyruvate is formed in the cytoplasm, and under aerobic condition it's to be carried to the mitochondria. Now it's through a mitochondrial membranes. Outer membrane is permeable, inner is impermeable, there are transporters, and then pyruvate is to be converted to acetyl coenzyme A. Our focus will be this pyruvate dehydrogenase complex and how pyruvate will be converted into acetyl CoA. And then of course there will be the Krebs cycle or citric acid cycle, and then these will be fed to electron transport chain for oxidative phosphorylation, and then there will be in presence of oxygen, ATP will be generated. This is overall picture of it. Our focus will be today on this part. So let us see in detail. This step is called as the pyruvate decarboxylation also, or it is also called as the formation of acetyl CoA, coenzyme A. Now I have already mentioned that pyruvate is generated in cytosol during glycolysis. We have already discussed this point. And since it is in the cytosol or in the cytoplasm, it's to be taken to mitochondrial matrix. It will enter it through specific transporters, which are called as pyruvate translocase, that allows the entry of pyruvate inside the mitochondrial matrix. And this pyruvate is then decarboxylated. There is a proper system of enzyme, multiple system of enzyme acting as a unit, which is called as pyruvate dehydrogenase complex, also named as PDH or PDC, which is in addition to this pyruvate PDH complex, it needs several factors, cofactors. We'll discuss it in detail. Then the net result of it will be, there will be the production of acetyl coenzyme A, 2-carbon molecule, reduced NADH, and carbon dioxide will be released. Now this reaction is irreversible, and this reaction is considered as a connecting link, or it is considered as a gateway step between the glycolysis and the citric acid cycle. It's a gateway point. We'll see what this pyruvate dehydrogenase complex is about. As I said, it's a complex enzyme. It's made up of three enzyme units, which are together. One is pyruvate dehydrogenase complex. It's also named as E1. Another second is dehydrolipoyl transacetylase, or it is named as E2. This is its molecular weight. And then the third one is dihydrolipoyl dehydrogenase. It is E3. And the 5 associated cofactors with this enzyme are thiamine pyrophosphate, TPP, lipoic acid, which is also called as a lipoamide. A lipoamide, let me tell you, it has 2 sulfur atoms, thus can donate and accept hydrogen atoms. And then there is a coenzyme A, FAD, and NAD. These are the 5 factors which are necessary for this functioning of the PDH complex. Now let us see step by step what are the reactions taking place in PDH complex, how it will convert the pyruvate to acetyl coenzyme. Mechanism of oxidative decarboxylation, as I said, it's a multi-step process. And you should remember, when there is a enzyme system involved, that time the intermediates of multiple step sequence do not leave the enzyme complex. So they are there. There's a channeling of substrate from active site of one enzyme to the active site of the next enzyme. This is the beauty of this complex. So there will not be intermediates produced. There will be an end product produced. And you will see this reaction, which is responsible for pyruvate decarboxylation reaction, is the overall, it's an irreversible reaction because it produces a free energy of -8 kilocalories per mole. That makes it highly irreversible step. This pyruvate will be converted to acetyl. It's an irreversible step. It takes place in 5 steps. We'll see them one by one. The first step is the decarboxylation. So pyruvic acid, which is a 3-carbon compound, in presence of an enzyme E1 or pyruvate dehydrogenase or pyruvate decarboxylase, and in presence of TPP, is decarboxylated. So there will be formation of carbon dioxide which will be evolved, and then there will be formation of this intermediate there, that is called as hydroxyethyl TPP, a 2-carbon unit which remains attached with the enzyme. And then of course there is a release of carbon dioxide molecule also. And then this TPP is basically a alpha B complex vitamin. Next step is this TPP acetate, which is hydroxyethyl group, is to be oxidized to form an acetyl group. It reacts with cofactor lipamide in presence of an enzyme 2 to form acetyl lipamide. We'll discuss it later on also. The enzyme, the E2, is involved, which is called as dihydrolipoyl transacetylase, and TPP is liberated. So this, along with the TPP, it combines with the lipoic acid. There is formation of this complex with the lipoic acid, and then TPP is released back for the next reaction to perform. Then there is one more that's called as a transacylation. Acetyl group from lipoic acid is released and combines with coenzyme A to form acetyl coenzyme A, which will be the end product of this reaction. Reduced lipoic acid is released. So we have this, it will combine with the coenzyme, reduced coenzyme A. In presence of an Lipoic dehydrogenase, there will be formation of acetyl coenzyme A. Of course, it will produce a reduced lipoic acid again. That reduced lipoic acid in the next step will be reoxidation of the lipoic acid. Then it will be with the enzyme, the third enzyme, dihydrolipoic dehydrogenase, where reduced lipoic acid, this enzyme has an FAD, a flavin protein. It will be converted to FADH2, and then there will be produced an oxidized form of the lipoic acid. Next step is the reoxidation of FADH. So this step 4, which we have already discussed, provides an oxidized lipoic acid back that is necessary for the formation of acetyl coenzyme from pyruvate. That's the next cycle. So regeneration has to be there. Lipoic acid is produced back. Then FADH2 reduces NAD to NADH plus H positive. This will be the outcome of this reaction also. That takes place in presence of an enzyme E3 or lipoate dehydrogenase. As I said already, it is a flavin protein, flavoprotein, which is tightly bound to FAD. So E3 catalyzes the transfer of electrons from reduced lipoic acid to NAD through FAD. And NAD, which is produced here, dissociates from the enzyme to be fed to the mitochondria. That is, this which is finally produced here, it is to be fed to the mitochondria for oxidative phosphorylation so that it will generate the ATP. Now we have seen one thing, that pyruvate binds at decarboxylate site, that's E1, and is converted to HETPP, hydroxyethyl thymine pyrophosphate. This enzyme, second enzyme E2 or transacetylase, contains two molecules of lipoic acid which are covalently bound to alpha amino group of lysine residue through an amide bond. Now the disulfide of lipoic acid is at end of the long chain which sweep over a considerable area and can reach to a catalytic site of adjacent enzyme. This is a very important point here. Disulfide group of oxidized lipoic acid picks up the two carbon moiety, forming an acetyl group, carrying it to the site of transacetylase subunit. So they are shifting from one to this. It is such a chain which has a disulfide at the end, and it can go over a considerable area. That is, it can have a connection, it can connect between the different catalytic sites of the complex. Acetyl group is transferred to coenzyme A, which is set free, and reduced lipoic acid is oxidized at a site on dehydrogenase to pick up another acetyl group from decarboxylase site. We have seen it in the reactions, and this was a summation of those. We can see it this way also. You have a pyruvate from the glycolysis that in presence of an enzyme E1 or pyruvate dehydrogenase, in presence of TPP, there will be formation of pyruvate hydroxyethyl TPP, and decarboxylation will take place there. And this will release TPP back. Now the enzyme 2 is coming in the picture. There is present a lipomide, which is oxidized, and it will combine with it. It will form an acetyl lipomide, and then acetyl lipomide, this enzyme will take coenzyme, and this will be released. Lipomide will be released, and with coenzyme A, it will form an acetyl coenzyme A that will be set free. While as this lipomide, which is reduced now, it will pass on its electrons to FADH2, and that to FAD to produce an FADH2, and then that FADH will result the reduction of NAD to NADH2. That means we have seen how these three complexes are working. What is the role of lipoic acid? Two lipoic acids are there. One is in an oxidized form, another is in a reduced form, and then the net product is this, and this is released, set free, and there is the release of carbon dioxide. This is how the three enzymes are working as a multiple unit. We can see this here also. You have a pyruvic acid from the glycolysis along with the TPP. This forms an HETPP, releases carbon dioxide in presence of enzyme 1 and forms this, and then TPP is released back. Then in E2 there is an acetyl lipoic acid formation, and there will be a combination of coenzyme, reduced coenzyme A, and it will be set free as a acetyl coenzyme A, and then this reduced lipoic acid will be produced back, but it will form the FAD reduced to FADH2 that will cause the reduction of NAD to NADH plus H positive, releasing lipoic acid in an oxidized state, and that will come again in picture with the next decarboxylation reaction. So that means we have seen this is a gateway step. Formation from pyruvic to acetyl coenzyme A, it's a gateway step. It is the resultant acetyl coenzyme A serves as a connecting link between glycolysis and TCA cycle for undergoing further oxidation. So these products which are produced, these reduced coenzymes which are produced here, cofactors which are produced here, they will be fed to electron transport chain for oxidation, oxidative phosphorylation, that will generate, of course, 3 ATP molecules from 1 NADH plus H positive, that we will see in the oxidative phosphorylation. But overall equation, what will be the overall equation? If you sum up it, these will be released back. TPP, lipoic acid all return to their original state. And so we have a pyruvic acid in presence of reduced CoA, in presence of NAD, 2 carbon molecules are given out. In presence of PDH complex, there's formation of an acetyl coenzyme A, 2 molecules, and 2 molecules of NADH plus H positive. This is the overall reaction of the conversion of pyruvate to acetyl coenzyme A. Gone through these, it has an end product inhibition. I have already mentioned it, which can act as a positive modulator, which can act as a negative modulator. And we have also seen that there are present 2 units PDC kinase, which will be an activated form or an inactivated form, that will in turn result in the regulation of PDH. And then we have also seen one more enzyme which is called as PDC phosphatase, which can be activated by high levels of calcium. As a result, it will remove the phosphate and will make PDH active. And hormonal regulation we have seen under well-fed condition, insulin will be produced, and we have seen protein phosphatase will be activated and PDH will become active. Under fasting condition, glucagon level will be high in the blood, and that is secreted by this pancreas, and there will be a protein kinase activated. As a result, that inactivates the PDH complex. As a result, pyruvate will accumulate and will be diverted to glucose by gluconeogenesis. Well, so far we have understood how the regulation of PDH complex is taking place. Let's explain it with this diagram. You will see this PDH, which is a multi-enzyme complex. This PDH is responsible for the conversion of pyruvate to acetyl coenzyme A, and end product is NADH plus H positive, which is a reduced NADH. So in this case, we have seen this enzyme PDH complex can be an inactive form or it can be in an inactive form. Once it is with a phosphate group, that time it is inactive. Once there is dephosphorylation, it is in an active state. So we have named it as dephosphoenzyme or as phosphoenzyme. So this PDH complex is the best example of feedback inhibition or the end product inhibition, wherein the end product, if accumulated, the end product here is the acetyl coenzyme A, and there is NADH, reduced NADH. This will act as a negative allosteric modulators for this complex. What will happen when there is an end product accumulation? That will have a negative modulation for this enzyme. As a result, it will get inhibited. Similarly, if there is an accumulation of pyruvate and reduced coenzyme, there will be this will act as a positive allosteric modulator for this enzyme, and there will be further conversion of pyruvate to acetyl coenzyme A. The beauty of this enzyme is that it has 2 complexes. One is called as a PDH kinase, another is PDH phosphatase, and this PDH kinase or PDC kinase is an enzyme which is responsible for the phosphorylation. This enzyme, if it is activated, it can be activated under 2 conditions. When there is more ATP, more NADH, more acetyl coenzyme A, that time this kinase will cause the phosphorylation reaction. It will remove the phosphate and will add it to the PDH complex. As a result, it becomes phosphorylated and it becomes inactive. So that means active PDH kinase will inactivate the PDH complex. And a situation can arise there. This enzyme, if it is inhibited, it can be inhibited under one condition, that is if there is a more NAD, more coenzyme, more pyruvate, more ADP, that time it will act as a negative modulator, allosteric modulator for this enzyme. It will be inactivated. As a result, of phosphorylation will not take place. There will be no attachment of phosphate, and this PDH will become as an active form. In addition to this PDH phosphatase, which is here, which is responsible for the removal of the phosphate, it removes the phosphate from PDH. As a result, PDH complex becomes dephosphorylated and it will become as an active PDH. This PDH phosphatase can be activated under one condition, that is if there is more muscular activity, there will be production of more calcium as a result of actin and myosin contractions. As a result, this PDH will be positively modulated. As a result, this will become active and will result in the removal of phosphate, and that phosphate removed will make this PDH complex as an active PDH complex. In case of human beings, a situation can arise under fasting condition and under well-fed condition. As we have already mentioned, under fasting condition, within this is a hormonal control of this PDH complex. Under fasting condition, there will be a production of more glucagon, which will get attached to the its receptors, and it acts as a positive modulator for PDH kinase. That means PDH kinase will be inactive. As a result, there will be no further conversion of pyruvate. Accumulation of pyruvate will take place there, and that pyruvate will be then reverted to glucose through the process of gluconeogenesis. That is when there is a fasting condition. But under this well-fed condition, there will be more secretion of the insulin by in the blood. That insulin, in addition to having the multiple metabolic effects, it will have a positive modulator activity for this PDH phosphatase. As a result, there will be removal of the phosphate, and that phosphate will removed from it will make this PDH active. And under this condition, when there is more insulin, that time more pyruvate will be converted into acetyl coenzyme A. So we have seen through this diagram how there is a regulation of this PDH complex which is taking place. This video will be quite helpful to you in understanding the gateway reaction, and we'll be discussing other reactions, especially citric acid cycle and oxidative phosphorylations in the next video. Till then, goodbye. Thank you.